Input is a small random vector. The output is so many smiles strings. What does it correspond to? It corresponds to generating several ligand molecules that are suitable for a given purpose. Okay? So you have semantic text vectorization. You have enormous amount of medical literature. So PubChem, PubMed, so much literature is there. So is there a way you can feed this data into the large language models that are there today? So given a protein molecule and a ligand molecule, can you synthetically generate some description about it? So this is one place called semantic text vectorization, where text vectors are fed into the system. I will show you some examples also. Translation. Can I take a text vector, my input is a text vector, maybe a query saying whether this drug molecule will it bind to the protein. That is my query. I want an answer which is also a text vector saying yes, it binds, no, it doesn't bind because of this and this reason. So how do I get such kind of input and output behavior? You have to first represent your data in the form of a vector, and the output of the neural network is also a vector which is then translated back to English sentence. So text to image. So you have this Meta app now. You give some imagination, you type some text saying someone is sitting in a room, and it generates an image out of it. So what is happening here is input text vectors are converted to image text vectors. So this uses a modeling called diffusion model. Okay? So irrespective of the mapping process, it's still a vector to vector mapping methodology. So region annotation, how can I mark a region in an image saying that these are all the various places that are of my interest? Imagine a protein molecule. A protein molecule is a three-dimensional image. Do you agree with me? It has X, Y, and Z coordinate as well. So in this three-dimensional representation, I can still represent it as a vector. Can I identify the binding site regions? The answer is yes. So the same methods what are applicable for marking regions in an image saying this is a chair, this is a timepiece, this is a fan kind of a thing. So that's called image annotation. Similar methods are applicable in this aspect as well. Those are all region annotation, but applied to three-dimensional vector of X and Y. 2D to 3D reconstruction. Given so many 2D photographs, can I reconstruct a 3D image? The answer is yes, it is possible. So all of these are possible from vector representation. So health monitoring. So I'm just giving a precursor or what is that requirement on using, before using any ML method, you need to be able to express your ideas in the form of a vector. So input is a vector and output is also a vector. So then ML tools can be used. So before the ML transformer era, a number of problems were of this sort, like applying linear regression on gene expression data. So you have tumor type classification, applying logistic regression again on gene expression type of data, on classifying the type of cancer tumor it is, applying GPCR families using support vector mission. Can I engineer a feature vector, feed it to the machine learning model, and get the output as what type of GPCR family is it? Is it glutamate, rhodopsin, and so on? Applying a decision tree for protein stability prediction. The input is a sequence level mutation. So I compose a vector corresponding to input, which is what mutation I'm having at a particular position, and the output I seek is the protein stable? Is the protein going to be stable or not? It's a binary classification problem. So here also, the input and output has to be designed in the form of vector. So protein-protein interactions. So how do I use a position-specific scoring matrix for as input to the ML algorithm. The output of it is saying in these locations, interaction is going to happen. So in all of these forms, vector representation was critical. So principal component analysis of gene expression. Can I cluster the gene expression data? Can I cluster the genes based on the lower dimensional projection of those genes? The answer is yes. If you are able to express all the gene expression in the form of a vector, you can calculate the eigenvectors and take some top 2 components of eigenvectors, project them and cluster them in that lower dimensional space. So these were the type of methods before the transformers changed the whole scenario. For example, this is also another example of clustering gene expression where you have short time series of data. You don't have only one snapshot, you have possibly 8 snapshots of the expression data. Can I cluster it? Cluster the time series of data. So can I construct a dendrogram and construct it from the gene sequences and to determine the sweep regions, the regions where mutations happen. So as Professor Prani told, biomedical image classification is one area where input is an image and the output is also a representation where you mark specific regions in the image. So input is an image representation represented as a tensor. Output is also an image representation represented as a tensor where you mark specific regions as it's a problematic area or not problematic area, what type of problem is it. So in all of these cases, human had to clearly define input and output behavior. So this was the area before transformers came into picture, where human had so much control on what type of data they want to engineer. So there was enormous amount of feature engineering where things were explainable because people hand compose those features. So if you feel that some aspect of the biological problem is important, you write a program for it, feed it as a feature to the machine learning algorithm. So now what happened, so after that what happened is things started to learn automatically from the data. That's called self-supervision. So it has advantages and disadvantages both. Explainability took a toll, but lot more problems were addressed using these transformers. So you also have this method called GAN, okay, generative adversarial networks. Typically it was used for low resolution to high resolution conversion. For example, if you take an image which is let us say 400 by 400 pixels and I want to make it 4000 by 4000 pixel highly clear image. So how do I do that? So the way to do that is to interpolate data, is to guess data. So if you plainly use any interpolation kind of a method, the zoomed in image will not be clear. It looks like blocks or splotches of image. So is there a smart way to do that? That's where this GAN formulation comes into picture. This is also useful in medical imaging. So where the input data is noisy and partially occluded, can I guess the intermediate regions and create a clear image? So GANs are not used in structural bioinformatics, but still it's one technology that we should know. So graph neural networks is another technology that is highly desirable for drug discovery topic. So you have these drug molecules. The task is to determine whether the given drug is active against a target or it's not active against a target without actually having to do the docking exercise. So docking takes time, isn't it? And if you are doing high throughput screening, so for every molecule you have to perform this docking exercise against the structure. So can I take a shortcut? The answer is yes. So the shortcut lies in this area called graph neural network, where you decide on a template of a molecule. Say that I'm going to have these types of atoms in my ligand molecule and create a grid representation of it. Can I find space fill the grid with certain molecules as being present or absent in the grid. So think of it like this. You have a white paper. In that white paper, you have so many cells that are there. You have a graph paper kind of a thing. So many cells are there. In each cell, you can fill what type of atom you want. You want a carbon or nitrogen, oxygen, hydrogen, different types of atoms you want, you can fill in there. So in this graph neural network, what you do is you select one type of atom in one XY coordinate of the graph paper, and then select another type of atom in the XY coordinate of the graph paper, driven by the data. Can I have a formulation that space fills the graph paper with what type of atoms I should put, what type of molecular fragments I should put. So this is one area of graph neural networks that also has picked up. So I will mainly focus on a couple of structural bioinformatics topics. So the big data here is the entire PDB, EMBL, all of this data is fed into the large language models that we have today. So we have about 140 large language models or transformer based models which were trained on this entirety of the data. So any types of questions, any types of text input, or the chemical input such as the SMILE string, the PDB input such as the structure representation, the machine learning models are able to map to the corresponding description of it. So let's look at the landscape of it. So RL, anyway, I will skip this. So let me directly show you this. So if you see, from 2018 onwards, this new technology called transformer has come into picture. So you have this GPT, BERT, Roberta, so many, you can see roughly about some 35 above types of architectures. There are so many sub-varieties, but only these varieties. So I have taken it from this paper actually, Ahmad Trian and others, 2024. So in this transformer architecture, the beauty is it is trained in a self-supervised manner. So I told you about the vector formulation, right? Input is a vector, output is a vector. Imagine that I have a sentence. In that sentence, first k words I consider as my input, okay? The next k words I consider my output. Is it okay? So starting from word I to word I plus k minus 1, you have k elements, that's my input. Starting from word I plus 1 to word I plus k is my output. So I create a ton of these pairs, so many pairs of x comma y from all of those databases what we have seen, PubMed, DrugBank, all of these databases. And I train a very, very large model having billions of parameters on these x comma y pairs. So what is it learning basically? It's learning to predict the next word. But is it how the human brain is working? We don't know. But the performance, how it is, what it is generating is similar to the human performance, what we are seeing in the LLMs here. So you have so many models. So I wanted to show you a slide where I have summarized, yeah, this one. So in bioinformatics, these transformers have directly been adapted to this domain called BioBERT. It started with BioBERT. You have these transformers for text. I will show you what is a transformer architecture now. You have this transformer for molecular data. You have it for protein data, genomic data, and multimodal data. All of these transformers are basically the next word predictors. So given one word, you want to predict the next word, but you train it on tons of data. So does it learn anything? The answer is yes. It learns the structure, structural representation of the data. It learns the semantics of the data. So if you see the text, the sources on which these transformers are trained were PubMed, PubChem, bioRxiv, arXiv, WIPO, and so on. There are about 12 transformers related to text, 12 types of architectures with variations in that. For molecular data, the type of data on which it was trained was Zinc, PubChem, USP patent, right? It's patent database. Then Geome, Drug Bank, and others. There are 40 transformers in molecular type of data on which are trained on that type of data. For protein, you have Uniref, Swissprot, and so on. There are 34 transformers. For genomic data, you have NCBI Genome. There are about 23 transformers in that area. Multimodal data, you have text-to-mol, drug chat, MOL-T5, all of those are LLMs, large language models. The chat GPT, what you are seeing, these are all that kind of models, where you can interact with the model with textual prompt. It will respond with the knowledge what it learned. So there are about 140 transformers applied in biological literature today. Okay? So all of these models, they make a lot of difference here. So what difference do they make? If you look at the protein structure prediction problem, AlphaFold won Nobel Prize in 2024. You remember? So these three people won Nobel Prize in 2024 for solving the protein structure prediction problem. Baker, Demis, and John. So what was their contribution? Their contribution was a transformer model for mapping a sequence to structure. What is the input? Input is a protein sequence. What is the output? Output is structure of the protein. So how did they do that? Okay. So the magic lies here. So the input was a multiple sequence alignment of the query protein. Query is a string, okay? So you take the query string, do a database of search for performing the multiple sequence alignment. You have thousands of rows, but a few hundred columns. The columns correspond to the amino acids of the query sequence. The rows correspond to the hundreds of sequences what you have aligned with. From there, how do we relate it to structure, right? So if you look at modeler, what does modeler do? Modeler takes a similar protein and tries to wind the query protein against the known structure, isn't it? So similar type of information should be captured here as well. So if we know the structures of the proteins with which the alignment has happened, we can take the coordinate information from them and then maintain the relative distances between the aligning residues. What do I mean by that? So if we take structure 1, it may be in some coordinate reference frame. If you take structure 2, it may be in some other coordinate reference frame. So how do we compare these two structures without actually having to align them? So one way to compare these two structures is, if you take pairs of residues, the pairwise distances should be maintained irrespective of the reference frame. Do you agree with me? Even if the reference frame changes, the pairwise distances would be more or less same if the structures have to align. So that's the trick used in this AlphaFold. So input is a pairwise matrix that has C channels. Channel means, think of it as an image. RGB is 3-channel image, right? RGB, width and height are the matrix dimensions of it. So here you have a C channel image corresponding to C proteins with which the alignment has happened. For every pair of positions I and J, you calculate the distance between the two coordinates and represent it in this matrix. You start with a random matrix and arrive at a refined matrix through multiple iterations. So it's like you start with a random structure, like the structure may be in any form which is not even matching with the known structure, but eventually the pairwise distances have to align. So there is something called triangle representation of the backbone. So take any 3 atoms, C alpha atoms in the backbone. So C alpha atom I, C alpha atom J, C alpha atom K, if you take some I, J, K atoms. So the triangle formed by the query protein, if it has to match with the database protein, this triangle should be similar, isn't it? Whatever final structure that comes later, this triangle should be more or less similar. So in this representation, for all triads of C alpha atoms, calculate a loss function. So is the loss value for the query protein matching with the triangles formed in the database of proteins, and try to minimize that loss over multiple iterations. So what is the input representation here? The input representation here is a vector again. So this vector has 2 components. One component is the multiple sequence alignment, which is coming. The second component is the pairwise distances between the C alpha atoms. So what is this about? If you see what is this whole pipeline about, is there is something called attention mechanism. So in the transformer, the critical invention Vaswani and others made was a small mathematical formula. Okay? So it's just one formula that made a lot of difference. So the formula was this. Let me show the formula here. Yeah? Only 3 minutes left. Yeah, that's fine. I'll stop. So I will show the formula of that. Yeah. You have the input represented in the form of a vector, am I right? The input is decomposed into 3 types of 3 names. The same input is decomposed into 3 names. This is called self-attention. You call it key, you call it query, you call it value, and calculate the interdependencies between key matrix, query matrix, and value matrix, and reduce the loss function to predict the value of a residue at a particular spot. Okay? So in multiple sequence alignment, what you have, you try to minimize the loss of predicting a given residue. So if you see here, you try to reduce the loss of predicting a residue in a given cell. If you look at multiple sequence alignment, each cell you try to predict by minimizing the loss on predicting correct amino acid is it predicted or wrong amino acid is it predicted. So this was the architecture that was used in AlphaFold. So AlphaFold has a database. AlphaFold, I ran it in my box also. I can show you some output. So I gave a query. So this is kind of a nice structure it has generated. Okay. So this code itself is runnable in the form of a Google Colab kind of a notebook. So the query protein was a sequence, like you can give any sequence you want, it will generate a structure. So one problem is it doesn't take your constraints into picture. So it will align the sequence to the database of sequences that it knows, but it doesn't take your constraints like binding site. This is the shape of binding site I want. This is the structure of the protein I want. So not yet, but still it is a quite useful tool. So I will stop here.